Assalamualaikum and a very good morning. A warm welcome to all participants from Malaysia, the SEAMEO network and affiliated countries from IMR-SEAMEO webinar series 2023. The theme today is Zebrafish model, a glimmers of hopes. Our presenters today are from the Institute for Medical Research in Malaysia. I'm Noriza Awang from Toxicology and Pharmacology Unit, Herbal Medicine Research Center, and will be the moderator for today's session. Toxicology and Pharmacology Units in Herbal Medicine Center conduct preclinical toxicology in vitro and in vivo on selected plants or herbal products. We run research on general toxicology, reproductive and developmental toxicity, as well as genotoxicity. Ladies and gentlemen, this webinar series was initiated with the aim to share knowledge, discuss topics on biomedical research, and present research outcomes. Before we begin the presentation, allow me to announce a few housekeeping messages from the Secretariat. We will not be taking questions during the presentation, but participants may post their questions using the link shared on the YouTube channel. The question and answer session will be conducted after both presentations have ended. An attendance link is also provided on the YouTube channel for those who would like to receive an e-certificate of attendance. Please subscribe to our National Institute of Health Malaysia YouTube channel and click for notification whenever there is a new post. Ladies and gentlemen, for today's webinar, we have two speakers. Our first speaker is Mr. Muhammad Rahimi Ashraf Abdul Rahman, who is a research officer in Toxicology and Pharmacology Unit under Herbal Medicine Research Center, IMR. He holds a master's degree of public health from the University of Newcastle, New South Wales, Australia. His research areas of interest are reproductive and developmental toxicity using an animal model. He is also part of the team members conducting research on the potential prenatal developmental toxicity and mutagenicity effect of Ficus retodia aqueous extract as well as Discoria hispida extract. As for today, Mr. Muhammad Rahimi Ashraf will be sharing his research interests with the presentation entitled Overview of Zebrafish in Toxicology Studies. Mr. Muhammad Rahimi, the screen is yours.
Assalamualaikum and a very good morning. My name is Muhammad Rahimi Ashraf bin Abdul Rahman, a research officer from Toxicology and Pharmacology Unit, Herbal Medicine Research Center, Institute for Medical Research. And today, I will be talking about an overview of zebrafish in toxicology study. First, what is a zebrafish? Zebrafish, with scientific name of Daniel are a type of freshwater fish that are part of the minnow family. They are native to South Asia, specifically in the stream of India, Bangladesh, Nepal, and Bhutan. Zebrafish are small, typically growing about 3 to 5 cm in length and have distinctive blue and silver stripe pattern on their body. They are omnivorous, feeding on various of small insects, crustaceans, and zooplankton. In the wild, zebrafish typically live for 2 to 3 years, but in laboratory settings, it can live to up to 5 years. Zebrafish are known for their regenerative capability, such as their fin, heart, retina, and spinal cord. Because of their genetic similarity in physiology to humans, zebrafish have become popular model organism in scientific research over the past few decades. Here are some of the brief history of the usage of zebrafish in research. In 1960, George Reisinger at the University of Oregon began studying zebrafish as model organism for studying developmental genetics. It is found that zebrafish easily raise in the lab and have short generation time, making it ideal for genetic study. In the early 90s, it begins to use in developmental biological research because of its transparent embryo. By the late 90s, zebrafish was found to be susceptible to range of human disease, such as cancer, heart disease, and neurological disorder. This led to an explosion of research using zebrafish. In the 2003, zebrafish's genome was fully sequenced and provided researchers with complete map of fish genetic codes. This enabled researchers to study gene function and regulation in zebrafish. Nowadays, zebrafish have become an important model organism to studying toxicology and drug development. So, the 3R value of zebrafish in toxicology. First, we have to understand the principle of replacement, reduction, and refinement in animal research were developed to promote more ethical and responsible use of animal in scientific research. First principle is the replacement. This principle was used to seek an alternative to animal testing wherever possible. This principle, the replacement is used to the zebrafish are often used to replace certain animal study, such as mice, in certain type of research. For example, zebrafish are commonly used in toxicity testing and drug screening. However, study validation for zebrafish to become fully utilized as model in animal need to be conducted. The second one is the reduction. The principle of reduction is to minimum number of animals necessary to achieve their research goal. As a first tier of toxicity, zebrafish could be used to identify toxic drug candidate, and it helped to reduce the number of animal use in mammalian model. And for the refinement the principle is to minimize the animal suffering and improve animal welfare. So zebrafish embryo were fertilized externally and often used in non-invasive procedure and observation. So why we use zebrafish in research? So zebrafish have become an important animal model research, especially in toxicology study. Our finding in the PubMed have shown that a total number of 9,001 number of publications are generated from PubMed when we are searching using a keyword zebrafish and toxicity. It is shown that it become a popular tools in toxicity testing and can be used in wide range of research area including genetics, developmental biology, neuroscience, disease modeling, and drug discovery. It is also ideal model organism for studying a variety of biological questions. So why we use zebrafish in research? First, zebrafish is known for its cost-effective and accessibility. The low cost of maintenance because zebrafish are small, hardy, and easy to care in the laboratory. They require relatively simple housing and feeding conditions, which make them less expensive to maintain compared to other mammalian models. Other than that, it is high reproductive capacity, which means it is rapidly reproduced, producing hundreds of eggs every week, which allow researchers to obtain large number of offspring for their experiment in relatively short amount of time. Other than that, it is easily to genetically manipulated and have wide availability. Zebrafish are wide available from commercial supply and other research institutions, which make them easily accessible to research around the world. This one is zebrafish also have genetic similarity to human. As it is fully sequenced genome, 70% of human gene have at least one zebrafish autolog, and 84% of gene known to be associated with human disease have zebrafish counterpart. Many of the new line can be repeatedly created by researchers through inbreeding, outbreeding, and transgenesis. This allows researchers to create specific genetic mutation or knockout for their studies. Other than that, most zebrafish organs perform the same function as their human counterpart and exhibit conserved physiology. This means that the response of zebrafish to toxic substance can be used to predict the potential toxicity of those substances in mammalian model and even in human. Other than that, its transparent embryo allow researchers to visualize and monitor the development of various organs and systems such as their heart, brain, eyes, and spinal cord, as well as the developmental of blood vessel. The real-time monitoring can help researchers to study physiological and behavioral response, providing insight into the mechanism of toxicity, making it an ideal tool for studying developmental biology and genetic disorder and the effect of toxic substance on different stages of development. Other than that, it is also used as an early screening to reduce the need of mammalian model, the high throughput screening of many compounds, and provided a cost-effective and ethical alternative. So now we proceed with the use of zebrafish in toxicology study. The first study is the acute toxicity that can be used on zebrafish. By following the OECD test guideline number 203 to determine the lethal concentration, zebrafish will expose for 96 hours, and the mortality and abnormal behavior and appearance will be recorded. The next toxicity testing is the embryo tox. By following the OECD test guideline number 236 to test lethality and developmental abnormality, there are 4 lethal endpoints that will be measured after the exposure to the test chemical for 96 hours, such as the coagulation of the embryo, formation of somite, and other than that. It will also become a popular model because of the externally fertilized and transparent embryo. The next study is the neurotoxicity and behavioral analysis. Zebrafish brain is much simpler than the human brain, but they have similar basic structure and function. Both brain have regions that control movement, sensory perception, and cognitive processing. This study will be later explained by my colleague in the later presentation. The next study is the cardiovascular toxicity. Cardiovascular physiology is conserved between human and zebrafish at anatomical, cellular, and membrane biological level. So many human drugs have shown comparable effect on zebrafish physiology. Zebrafish, furthermore, can regenerate their heart muscle. Therefore, researchers are now working on factors involving in this process to help to develop method of repairing heart tissue in human. The other study that can be conducted is the hematologic toxicity. Zebrafish also have many blood cell types that consistent with human peripheral blood cell, such as red blood cell, neutrophil, monocyte, macrophages, T and B cell, and dendritic cell. This can help us to determine what is the effect of toxicity in the product to the blood. Other study includes intestine, pancreas, and hepatotoxicity. The zebrafish liver perform the same function as those in human liver and is fully functional by 5 days post-fertilization. Larva zebrafish can be used to integrate xenobiotic from hepatotoxicity in vitro format, provided whole organ or animal data. The next study include the nephrotoxicity. Despite having a simple anatomy with 2 nephrons with fused glomeruli called pronephron, the similarity between the cellular constitution and function between mammal metanephron and zebrafish pronephron make a room for nephrotoxicity studies. Other than toxicity study, the larva zebrafish also are being used in phenotyping screening to discover compound with a potential to repair acute kidney injury. The next study include the ocular toxicity. As we know, the zebrafish eye is highly conserved with gene expression, cellular makeup, and tissue architecture to human. An obvious anatomical similarity include the fish also have cornea, lens, choroid, and retina. Because of that, researchers use it as a model to investigate pharmaceutical ocular toxicity, especially reaction to drugs. And the last study include the ototoxicity. As we know, a large part of our understanding of the genetic of human hearing and balance have come from study with zebrafish. Zebrafish screening for autotoxicity in mammalian preclinical model are very difficult, and given few regulatory requirements, this is rarely done. A zebrafish model for autotoxicity may fill this gap in preclinical testing. Latest advance and future direction of using zebrafish. By invention of CRISPR-Cas9, gene editing to create mutation in zebrafish gene. This helped researchers to create specific mutagenic zebrafish that help to study the gene function and disease mechanism in detail. Other than that, the optogenetic also have been used, which include the technique to control the activity of specific neuron or other cell type with light. This can help researchers to study the heart, neurocircuit, and behavior, providing insight into the underlying mechanism of heart and brain function. Other than that, the development of advanced imaging and automation technique help researchers to create detailed and high-resolution 3D map of zebrafish embryo, providing insight into the complex processes of development. The advanced automation equipment and data collection also can become more faster and accurate to provide more reliable data for researchers. And last, by the advance of technology and imaging can help drug discovery setting, which will help researchers to screen more potential drugs. To screen potential drug candidate for efficacy and toxicity, allowing them to identify promising compound. Looking to the future, zebrafish research is likely to continue to become more important contribution to our understanding of developmental, genetic, neuroscience, and disease mechanism. As technology advances, researchers will be able to study zebrafish even greater detail, providing new insight into the complex biological process that underlie life. From our unit, toxicology and pharmacology unit, we have just established zebrafish lab early this year. Also, in the early this year, we also have received various visitation from other companies such as Institute for Infectious Disease Sarawak, visitation from students from College Mawaib, and is included secondary student from SMK Alam Damai. This is some of the research we be able to capture using our stereo microscope in the lab. From this slide, we can see that the eggs were collected the 0 hour post-fertilization in the morning. So in this slide, we can see that the fertilized and unfertilized embryo, which later will be sorted and taken for before any treatment can be conducted. After the treatment be conducted, we will be analyze the potential teratogenetical defect that happened at the embryo. From this one, we can see that the eye development, the somite formation, and the tail detachment, which is one of the criteria for mortality endpoint. At the 48 hour, the pigmentation start to produce and the heartbeat start to pumping the blood all over the bodies of the zebrafish. And at 72 hour post fertilization, the zebrafish start to hatch from their chorion, and this is the picture taken 96 hour post fertilization of the zebrafish. So from this slide, we can see also the imaging, the video we capture when studying zebrafish. At 24 hour post fertilization, we can see that the movement of the embryo will help us to determine and to calculate the heartbeat of the embryo. At 48 hour also, we can see that the heartbeat and the flow of the blood circulation in the zebrafish, which also one of the main criteria to observe in our studies. In the 72 hours post fertilization also, we can see the zebrafish hatch from the embryo, and we will calculate the time for the zebrafish to hatch. And this last video, we can see the blood circulation, the pumping of the heart, and the blood circulation throughout the all the bodies of the zebrafish. That's all from me. That's all from me. Thank you for listening. Back to Puan Noriza.
Thank you, Mr. Muhammad Rahimi, for the interesting talk. If there is any question that you would like to ask about his presentation, kindly submit your question using the link provided in the YouTube channel. Next, I would like to introduce our second speaker for today, Mr. Pu Chinlong. Mr. Pu like to conduct research on cancer, traditional complementary medicine, as well as neuroscience. He was involved in a research project entitled A Systematic Approach to Study TCM as an Adjuvant Treatment in the Breast Cancer Patient. He had an internship at National Health Research Institute, Taiwan, with a research project neuroprotective effect of Topamax against ischemic stroke in rat model. It is our great privilege to have Mr. Pu Chinlong with us to share his background of research. His presentation title is Zebrafish as a tool to assess neurotoxicity. Over to you, Mr. Pu Chinlong. The screen is yours.
Thank you, Puan Noriza. A very good morning to the audience. I'm Chinlong from Toxicology and Pharmacology Unit. Today, I will walk you through my research interest on the title of Zebrafish as a tool to assess neurotoxicity. So what is neurotoxicity? Neurotoxicity is defined as an interaction of chemical substances with the nervous system, primarily leading to structural changes, destruction in structural and cellular synaptic function that result in variety of neurological symptoms. The potential mechanism of neurotoxicity includes oxidative stress, inflammation, autophagy, and apoptosis, as well as axonal degeneration. Neurotoxicity can result from the exposure to environmental toxicants, pesticides, insecticides, heavy metals, drugs used for chemotherapy, radiation treatment, and organ transplantation, herbal medicines, as well as food additives. Testing for neurotoxicity has becoming more and more important due to the increasing exposure to neurotoxicants, especially environmental toxicants. This can induce significant risk to human health and cause different neurological impairments and disorder. Current neurotoxicity assessment is always based on in vivo experiments conducted in rodents. One of the main neurotoxicity endpoints measured in rodents is behavioral or functional assessment, which include motor activity, motor function, sensory function, and learning and memory. This table shows different neurotoxicity endpoints with their respective tests. For example, we can use open field tests to assess motor activity, grit strength for neuromuscular weakness, and rotarod test for incoordination. Both these tests can use to assess the motor function of the rodents. Besides behavioral assessment, neuropathological alterations, neurochemistry, and electrophysiological findings are other examples of neurotoxicity endpoints that can be measured in rodents. As you can see, there are many neurotoxicity assessments available in rodents. However, all these tests are laborious, time-consuming, and not suitable for large-scale screening of chemicals. Therefore, an alternative model, which is Zebrafish, is used to study neurotoxicity. Zebrafish offers many advantages, such as high reproductive rate, external development. It has high genetic similarity to humans. Besides, the optical transparency of zebrafish larvae allow us to visualize the neuronal activity and neuronal structure easily. In addition, the structural organization and cellular morphology of zebrafish brain is very similar to other vertebrates, including human. Zebrafish also exhibit a wide range of complex behavioral pattern that is highly comparable to humans. Neurotoxic effects of chemicals can be evaluated by different neurotoxicity endpoints, including gene expression, neuromorphology, and neurobehavioral profiling. The method used to detect the changes in gene expression are in situ hybridization, qRT-PCR, microarray, and RNA-seq. Neuromorphogenesis can be visualized by immunohistochemistry, acridine orange staining, and use specific transgenic lines of zebrafish. Neurobehavioral profiling is one of the most commonly studied neurotoxicity endpoints in zebrafish. The examples include locomotor activity, social behavior, anxiety, and learning and memory. These are some of the examples of studies done on zebrafish which measure gene expression as their neurotoxicity endpoints. In this study, qPCR was used to determine the expression levels of 9 marker genes that are related to neurodevelopment and nervous system functions. In this study, acridine orange staining is used to evaluate the neuronal apoptosis in treated zebrafish. The apoptotic cells can be reviewed by the AO staining, and the presence of apoptotic cells in the brain indicate chemical-induced apoptosis in nervous system. Transgenic zebrafish embryo that express green fluorescent protein GFP from the axon of facial sensory neuron and peripheral motor neuron were used to assess the axon damage and peripheral motor neuron damage as shown in the diagram. Immunohistochemistry, IHC, is used to visualize the dopaminergic neurons as indicated by the tyrosine hydroxylase TH positive cells, primary motor neuron PMN, and secondary motor neuron SMN. Next, moving forward, this is the overview of some of the neurobehavioral assays currently in use with zebrafish that is assigned to different timeline of development in zebrafish. For example, at 17 hours post-fertilization (HPF), zebrafish starts to show spontaneous contractions, followed by the touch response at 21 HPF, with the tail coiling to a tactile stimulus given to the head. On day 3 to 4, locomotor activity and startle responses can be measured. There are also other neurobehavioral endpoints that can be measured in zebrafish from 4 DPF onwards, such as rheotaxis, phototaxis, automotor response, and autokinetic reflex. So these are the examples of neurobehavioral endpoints measured in zebrafish. First, the touch evoked response test is used to track the behavior of zebrafish in response to a tactile stimulus given to the head or the tail. The touch evoked response is an indicator of the integration of sensory and motor function. Second, the startle response can be induced by different stimuli such as vibrational stimuli or acoustic, visual stimuli. For example, the vibrational stimuli can be elicited by tapping the multi-well dish. As shown in the diagram, the normal startle response is characterized by the C-shaped band of the body as shown in the control group as compared to the treated group. The third neurobehavioral endpoint measure is the locomotor activity. It can be measured by photomotor response and alternating light dark locomotor response assay. Briefly, the zebrafish will be placed in a system chamber which is equipped with an automated video tracking system. There are also many anxiety test models in zebrafish, such as open field tank, light dark test, and novel tank diving test (NTT). So this NTT exploits the natural tendency of the zebrafish to dive to the bottom of the tank and gradually exploring the top zone of the tank. So the normal zebrafish typically explore the new environment, the top area or the shallow area of the tank, while the zebrafish with anxiety will tend to swim at the bottom of the tank. Next, the predatory avoidance test is a behavioral response in zebrafish that occurs when the predators are nearby. So the zebrafish will freeze and retreat when they are from their predators, as shown in the diagram. Next, the learning test, for example, T-Maze test, is a behavioral essay that measures the exploratory behavior. This test is based on the willingness of the zebrafish to explore a new environment or novel environment. The zebrafish prefer to visit the new environment of the arm of the maze rather than the familiar arm of the maze. So they will tend to swim to the new arm of the maze. Lastly, the social interaction test can be divided into shoreline tests, which assess the behavior of zebrafish in the presence of a group of conspecific fish that is the same species of fish. And the next one is the social preference test that assess that studies the behavior and interaction of fish indicated by transparent barrier. So this is an example of study done on locomotor activity of zebrafish after treatment with a food additive. So in this study, alternating light dark locomotor response assay was used to determine the locomotor activity of treated zebrafish larvae. So the parameter measures are the total distance traveled, the average speed, and the locomotor pattern of zebrafish. So in conclusion, the advantages of zebrafish and many neurobehavioral endpoints discussed earlier make zebrafish a useful and potential complementary model to classic rat models for studying neurotoxicity. The advantages, especially the compliance with 3Rs, replacement, reduction, and refinement of animal studies, would ultimately reduce the number of animals used in testing. However, there are still some limitations and challenges for studies of complex behavioral changes in zebrafish. A systematic comparison of chemical neurotoxicity in zebrafish and mammals is necessary to validate zebrafish as an alternative model for neurotoxicity. Thus, zebrafish can be used as a screening tool to assess the toxicity of drug candidates. That's all for my presentation. Thank you for listening. I will now pass back to Puan Noriza.
Thank you, Mr. Pu Chin Long, for the insightful presentation. We will now be moving to the question and answer session. Please do not hesitate to send us your question using the link provided on the YouTube channel. And I will try my best to connect you to our distinguished speaker through the posted question. Nevertheless, should the time not allow us to address all the questions, our speaker will try their best to answer the question through email. It seems that we have received a few questions from our audience. A question to Mr. Pu Chin Long. What is the limitation and challenges so far in conducting zebrafish research?
Okay. Thank you, Panorisa. So for this question, there are a few of limitations and challenges when we conduct the zebrafish larvae. So in specific to neurotoxicity, we have to know that there's no single animal model that can target all the brain complexity that is happen in human brain disorder. And there's still substantial differences in the brain complexity in zebrafish as compared to humans. And the CNS structure, the brain, some of the brain areas are not as developed as in zebrafish as compared to humans. And the next limitation will be the water solubility of the test compounds. So if the test compound is not water soluble, it will be very hard to administer through the water dosing, the water immersion. So it has to be injected. It has to be injected to the fish. And also, because for zebrafish, we need to use the water immersion of the test compounds. So the compounds that is actually absorbed to the zebrafish cannot be determined. So So the concentration or the dosage might not be accurate as stated.
There's another question from Shazlan. Where do you see yourself in 5 or 10 years as zebrafish researcher in your research field? The question is for Mr. Rahimi.
Okay, thank you, Reza and Shazlan for the question. For the 5 to 10 years, so we will be hoping in our unit to become an expert in zebrafish study. Also, in our research center, which is Herbal Medicine Research Center, we also, there is a lot of herbal that still unknown, their toxicity effect and maybe their therapeutic effect on human and maybe on mammalian model. So we are hoping that we can use this zebrafish as a model, also as a first year toxicity studies to help other researchers to find the toxicity effect of certain herbs or any extracts, herbal extract, so that we can further our study, a future study into a preclinical and clinical trial.
Okay. There's another question for Mr. Rahimi. Which development?
So I think the embryo stage typically are very easy to use, easy to obtain. The egg also easy to obtain, and as I mentioned in the previous slide, the transparent embryo make us as researcher easy to get more reliable data. So we can have we have very proper timeline to observe the developmental progress of zebrafish, and the embryo also provide a data that can be crucial, reliable, and more accurate data that we cannot lie in the publication and maybe in the presentation later on. Yeah.
Okay, this question for Mr. Chin Long. You did mention of rheotaxis and phototaxis in your slide. So what is actually rheotaxis and phototaxis?
Okay, thank you, Panarisa. So rheotaxis, yeah, I did mention the rheotaxis and phototaxis. So rheotaxis is actually used to detect whether there is an impairment in the hair cells of the zebrafish. So the normal zebrafish will orient themselves and swim towards the normal flow of the water current. So if the larvae after treated with the chemicals, if there's an impairment to their hair cells, they will not able to orient themselves to the water current. So normally the test will be conducted in the fixed water tank. So there's a continuous water flow to the zebrafish. And whereas for the phototaxis is used to determine whether there's an impairment to the vision of the zebrafish. So the zebrafish will be placed in a chamber. There's a light dark box in the light dark box. So the normal zebrafish will tend to move to the illuminated one, which is the light box. If the zebrafish move to the dark one, then that indicates there is an impairment to their visions.
Okay, thank you, Mr. Chin Long. A question to Mr. Rahimi from Noor Amira. Have you ever succeeded to breed the embryo to a darkfish?
Okay. Very good question from Norma Amira. So right now our lab are having some difficulties, and that difficulty is sometimes is try to breed the embryo into the adult zebrafish. So right now we have succeeded in mating 5 mating session for our experiment, but to breed until the adult, we still haven't figured procedure to do that. We are trying. Right now we are trying because the last we have success mating zebrafish in I think last 2 weeks, and the zebrafish right now is 20 days post fertilization and still alive. But in the laboratory session, maybe from 100% from we use percentage from 100% we successfully to mate. We have problem maybe right now only live maybe about 60% that live through the 20 days for post-fertilization. So right now we are having problem. Maybe we try to identify some of the problem. So the previous one is we find it difficult to feed the fish using the powder provided by our supplier, and now we are trying to feed the zebrafish using live using artemia, to look whether it can pass the 20-day post fertilization. So right now we only manage the longest we manage to breed until 30 days for post fertilization. So right now we are trying to figure out the problem and step to do how to breed the zebrafish from larva into the adult stage. Yep.
Thank you, Mr. Rahimi. There's another question for Mr. Chin Long. Is there any OECD guideline for neurotoxicity testing?
Okay, thank you, Puan Reza. Yes, there are two OECD guidelines now. So the first one is OECD 424 guideline, which is the neurotoxicity in rodents. So basically, this 424 OECD guideline is the guideline that we can use to confirm the neurotoxic effects usually conducted in adult rodents. And there's another OECD guideline, which is 426, that is developmental neurotoxicity. So in this 426 OECD guideline, it is usually conducted in pregnant or lactating female rodents. So this 426 studies is usually incorporated in prenatal and reproductive toxicity testing.
Okay, thank you, Mr. Chin Long. There's another question for Mr. Rahimi. Can this model replace the existing animal model for research? Mr. Rahimi?
Okay, can this model replace the existing animal model? Okay, zebrafish alternative still not considered as true in vivo system by regulatory bodies because they still demand a mammalian model and human and clinical trial data. But as I mentioned in the slide, it can give us insight into potential therapeutic and toxicity effect and help the specific or the right compound to be tested on mammalian model and even in human.
Alright, I just would like to add OECD guideline as mentioned by Rahimi's slide. For the OECD guideline, 203 for acute tox and OECD 236 for embryo tox. So I guess the Malaysia regulatory have to look at to accept this as to be used in Malaysia for the data for the to support the clinical study, because OECD guideline is using it for acute tox. So maybe we can replace the rodent for the acute tox in the future. Yeah. So we have another few questions. Let me scroll. A question for Mr. Chin Long. What is the difference between a photomotor response and alternating light dark locomotor assay?
Okay, thanks, Puan Reza. So for these two tests, so basically these two assays are used to assess the locomotor activity. So the main difference is that the photomotor response is a non-visual response, whereas for the alternating light dark response is a visual response. So the photomotor response is usually conducted in pre-hatching zebrafish, whereas for alternating light dark response is conducted in post-hatching zebrafish. So for the photomotor response, it's actually, since it's the non-visual response, so the light is actually mediated by the photoreceptors in the brain of the zebrafish, whereas for the alternating photo response, it's actually used to determine the locomotor activity of zebrafish when it is eliminated to alternating light dark period. So yeah, so these are the differences for these two assays.
Next question, another question from Ahmad USM. The question is the zebrafish model available for university researchers? If yes, what is the procedures and expectations? And how much does it cost per fish? Mr. Raimi, maybe you can answer the question.
The Jilong can also answer because he purchased the zebrafish. So for IMR right now, we are still trying to, as we mentioned earlier, we still try to breed it into adult stage. So the adult we still cannot provide, and also we still have to develop a proper procedure to enable us to sell, maybe to sell the embryo and the adult fish. So right now we are still not doing that, and usually cost per fish around 3 ringgit per fish, 2 to 3 ringgit per fish. If from what we are purchased early this year. Yep.
Okay, maybe we can collaborate. Yeah. Okay. Let me scroll for the question. Question to Mr. Chin Long. What are the example of neurotoxic effects in humans and animals?
So there are many neurotoxic effects or neurological symptoms in humans and animals. So, for example, the solvents such as hexane and acrylamide, they can both cause ataxia, tremor, and paralysis in humans, and it can cause weakness and loss of fine motor control in animals. And other examples will be the heavy metals such as the arsenic and mercury. Both can cause hyperemesis in humans and animals. Yeah.
All right, thank you, Mr. Pu Chin Long. There's a question to Dana. Let me scroll. There's two questions from City MOH. Is the efficacy data obtained from zebrafish study always applied to small rodent? For Mr. Raimi, can you answer this?
Okay, the efficacy data obtained from zebrafish study can be used and can be applied to the small rodent. And do the IMR offer safety study using zebrafish in JLP? I think not yet. Right now it's no, not yet, because zebrafish, as we mentioned, still not accepted in especially in Malaysia regulatory body.
This was question from Siti Aminah from Agensi Nuklear Negara. Her question is, for Alzheimer's study between rodent and zebrafish, which one is more preferred to use? Chin Long, can you answer this?
Yeah. So both rodents and zebrafish actually have their own advantages for this for Alzheimer's study, but I would say for the Alzheimer's study, for the efficacy study, it will be rodent, and for screening tests, it will be zebrafish. So there are actually a variety of tests available in both models, rodents and zebrafish. So for zebrafish, because of the small size, is more easily to be manipulated as compared to the rodents.
Yeah, if you ask us in doing the with the zebrafish facility, of course we prefer zebrafish because easily be handled compared to rodent. Yeah.
Yep.
There's another question from Dr. Syah Kamal. Can you use zebrafish as a model to study tumor development and the efficacy of cancer drugs? If can, what are the parameters to evaluate drug efficacy? Question to Rahimi. Can you answer this?
This one difficult question. Uh, to study the tumor development, yeah, I think we can study using zebrafish because of the gene. We can manipulate gene in zebrafish to 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 alter the genetic of zebrafish that can be used in tumor study. The parameter, I think I will get back to you later. Because yeah, this is beyond my expertise right now because we just are still learning. We are still research. Still to develop a method for toxicity study using zebrafish and because like I mentioned earlier, the lab just established early this year and from that we are learning to conduct a lot of other toxicology and the efficacy of the drugs using zebrafish.
Yeah, can I add some more? For the drug efficacy, I think you can look at the gene expression, whether it's overexpressed or not, and you can target the gene expression for the to see whether the drug is effective or not. There's another question. Aside from neurotoxicity, do zebrafish can be used to study endocrine disease? Chin Long, can you answer this?
Yeah, I'm sure it can be used to study endocrine disease and also other diseases. Yeah, apart from neurotoxicity, there are many other potential fields for zebrafish, not limited to neurotoxicity.
Yeah. Let me see if there's other question for us to answer. Question to Rahimi. How to differentiate between female and male zebrafish?
Okay, at our facilities, right now we have ways to differentiate female and male zebrafish. Usually, female are much smaller than male are much smaller than female zebrafish, and the shape of the female more flat compared to female because female the belly is quite big because they contain eggs. They contain eggs for mating, and other than that, the male usually darker in color compared to the female zebrafish.
Okay, thank you. Last question is for Jin Long from Muhammad Hamzah Mat Nasir, IIUM. The question is, for neurobehavioral tests, do you need a quiet room with control lux, or you can just use the maze in an open lab? Or perhaps we can collaborate as we are working with Parkinson models as well as CRISPR for neurogenesis study.
Thank you, Dr. Hamza, for the question. So for this neurobehavioral test, for this T-based test, as I mentioned, it is used for the learning test. So I'm sure the light-dark cycle, cycle is a would be a confounding factor, a main factor for this study. So yeah, we do need a quiet room. So the T maze, the setup will be in the quiet room with the control light-dark cycle. And yeah, we are looking forward to collaborate with you in the future.
Okay, I am afraid that is all the question. That is all time we have for question and answer session. Any additional question will be answered by speaker through email. We hope this webinar has shed light on some research work that we are conducting in IMR and would spark interest and could initiate research collaboration in the near future. As for toxicology and pharmacology unit, we have established the Zebrafish facility and we are open to research collaboration with others. Ladies and gentlemen, now we have come to the end of the webinar session. I would like to thank all participants for their support to make this session successful. Don't forget to subscribe to our National Institute of Health Malaysia YouTube channel. Before I conclude, I would like to announce that we will have the next session of the IMR-SEAMEO webinar series on the transplant puzzle, joining the pieces together. So mark your calendars. We hope you can join us at the next session. Until then, many thanks from all of us at IMR. Take care and stay safe.